everybody. On behalf of the Society of Thinking House, I welcome you all on the third and final day of the Popli Memorial Lecture Series. Before we begin, I would like to step down to the mobile phone to the right side of the remain in the lecture lectures. Throughout the last few days, Dr. Prerna Sharma has taken us on an incredible journey through the physics of waves. We have explored the physics of biomemory, vesicle formations, and thermal fluctuations, as well as the dynamics of molecular molecules and the active cytoskeleton. She has shown us how the beauty of physical laws underlies the influences of life itself. As we begin the final section of the lecture series, we look forward to the concluding chapter. Without further ado, I invite Dr. Prerna Sharma to deliver the final lecture of the 50th Popli Memorial Lecture Series. What happens when you start thinking about these things differently and writing down equations for that? So, just as we had done in the last class, what you can do is you can write down in a small region around plus n, there are n plus monomers, and great happening at the two different ends, and you will get two critical concentrations, which are the ratio of the two rates. So now there are, so remember that I mentioned to you that microtubules, so there are two two filaments that are really important in the cytoskeleton, microtubules and actin, and we can, and what we get is, like from these late equations, two kinds of behavior. So for microtubules, this kind of behavior appears where the two critical concentrations are the same. And what you will see is that what this graph tells you that both rates will either grow simultaneously or shrink simultaneously. Also, the rate at which they grow or shrink can be different. So the slopes of these curves are different, but the regimes of growth and shrink are cooperative. So that's the case for tubule. For actin, this situation is more complex, where you have the two critical concentrations are different, and so you have these three regimes. Where below this critical concentration, both ends will shrink because VN by 3 is negative, and above upper critical concentration goes to 0. But there is this intermediate regime where both, one end will grow and the other end will shrink. And you can see that at this concentration, so there is a unique concentration where the two rates can have same slope. So at end steady state, you find that the local slopes are very similar, and then you get into a situation. So when you have something like this, you will get what is called treadmill, in the sense that, so one end is growing and the other end is shrinking, but the rates are the same. So the overall filament length is the same, but then what this will do is something like this, that you are having one monomer attached and then one monomer come off. But this highlighted monomer is actually now moving, through the film. So this is what we mean by treadmill. So why this is important in biology is because you can have proteins that can bind to these tracks, and when this track is undergoing a treadmill like this, then that protein will have a direct diffusion. So light is always trying to engineer ways to have directed motion, and this is one of the ways to engineer. And then, so this is the uniqueness about actin. And the microtubule also has a very interesting instability associated with it. So what people found is, so here is the length of a microtubule as a function of time. And what they find is that despite having, a constant soup of monomers, this filament will grow and then abruptly about these ends being equivalent, etc. But at the same, in addition to that, there is a process of hydrolysis going on. So when the monomer attaches, it gets hydrolyzed and takes a different form. So let's think of it as a red thing getting converted into a blue thing. And as long as you have the red cap, it's a very stable filament, it will keep on going. And what is happening is that these red things are very stable, so they don't fall off. But if the blue thing becomes exposed, then the blue thing will fall off very quickly. Like depolymerization takes a high. So when you have lots of monomer, then this cap can be very long. So essentially there are two rates. There is rate of attachment and then there is rate of hydrolysis. So the rate of hydrolysis is the rate at which the red gets converted to blue. So if the rate of attachment is much higher than rate of hydrolysis, then you develop this very stable cap and you have a steady growth. But if there are monomer fluctuations or the monomer concentration is low, then the two rates kind of catch up and you no longer have a very stable end. And as soon as some new monomer get exposed, then it will polymerize dramatically. And that's how this rescue catastrophe continues to happen. So the point of all this exercise was I wanted to show you that how biological polymers can be very rich in phase changes. So synthetic polymers would never do something like this. Because synthetic polymers are driven by this completely boring and enthalpic interaction. Something comes and you have a reaction and it binds and that's about it. But here you have all these mechanochemical processes happening that these polymers can make use of. And the reason the cell would want to have something like this is the following. That this is one of the ways, but what the cell is really trying to do is it's trying to set these filaments like a probe. So the cell wants to probe in the boundaries, for example, and so on and so forth. So you have a microtubule that's growing in some direction, and let's say it has found the target. Now, since the target has been found, you want to do something else, or you want to retract. So retraction can be very fast because I don't really care. I go slowly because then I have the thermojoids to give me the advantage of finding targets. But as once I found the target, then I can quickly retract. So this dynamic instability of microtubules is beneficial for the cell in this way. What you are looking at is a dividing cell, and there are these microtubules, and these blue things are the chromosomes. And here there are a lot of monoproteins at work which slide the microtubules, and you can really see that they can. So this assembly is called spindle. So it's basically like a pneumatic droplet of these rod-like systems, and then you have a lot of motors that kind of pull on this droplet, and in that process it segregates the chromosomes. And then, as I was mentioning to you yesterday, that you don't necessarily need motors to generate forces. Even simple polymerization is good enough. So here you have a vesicle in which these people glue microtubules inside. So what you can see is that you started out with a spherical vesicle, which then really gets extended into ellipse, and then an ellipse with tubules extending out. And that's mainly because the microtubule was growing. So simply polymerization is good enough to exert forces on the membranes. So now I will stop. slide on how liquid binding was formed, etc. But the thing is that so as I mentioned to you, these membranes are very thin, right? They are 5 nanometers in thickness. So experimentally, it can be very challenging to visualize them. Even though I showed you movies where you could see their large scale fluctuations, etc. But if you are really trying to measure forces or look at micro scale deformations, then it becomes challenging because your posterior lens scale is 2 nanometers. So you cannot use standard optics to like visible optics. So there is this keen and soft matter where you say that you can map what's happening in atomic crystal to what's happening in a colloidal crystal. So colloids, as I mentioned to you in the first lecture, are these micron-sized particles, and you can engineer interactions in them in such a way that they will self-assemble into a crystal. And the idea here is this crystal is macroscopic. The lattice constant will be few microns. So you can easily use an optical microscope, visualize the structure, visualize the dynamics, apply forces, and learn a lot, right? And you can have a mapping to what happens in the atom crystal because finally you are thinking about pair potential. So if you can have a similar pair potential in the colloids, then you have the same physics, so to say. And the work that we were doing is we wanted to have a similar mapping for the case of binaries. And so essentially what I wanted to do was to take a sheet, sheet-like structure, but whose thickness is about a micron. And the way, and these membranes should satisfy these hallmarks of real membranes, which is two-dimensional, fluid-like dynamics, long-lived elasticity, and simple enough to analyze theoretically and tune experimentally. So one of the, so the building block of these model membranes is rod-like virus. So this is a virus which is 1 micron in length, few nanometers in diameter, and it is highly charged, etc. And so you can see this virus, it looks like a rod, but it's not a perfect rod. It's a little bit semi-flexible rod. And the reason, and this virus is a bacteriophage, so it infects bacteria. So it's very easy to prepare in the lab. You grow lots of E. coli, infect it, and then E. coli will multiply the virus for you, and then you end up with a soup of rods which are like nature has prepared for you, right? And all these rods will have the same length and same value because what is a virus? It's a piece of DNA coated with proteins. So as long as the DNA stays the same, we are going to have this templated rods with you. And making nanorods of this high aspect ratio, 1 micron in length and 6 nanometers in diameter, is still chemically not possible. I mean, you might be able to make it, but to have a monodisperse suspension, by monodisperse I mean all rods the same length and same diameter, is highly challenging. So we use this. So basically here I am using a biological material as just a column. And what we can do is we add polymers. So now schematically you are looking at virus as these red sticks, and these blue coils are the polymer coils, and these polymers will apply osmotic pressure and bring the rods together. And if you have many such rods, you will end up with these small little sheets that will can coalesce sideways like 2D droplets, and you can have these really large sheets which are tens of microns in diameter and 1 rod length thickness. So this is a model system that I have worked on for many years, and this is how a good sample will look like. So under the microscope you will see lots of these sheets, and these ones are edge-on, so they appear fixed. Here I have shifted to fluorescent, where 1 out of 10,000 rods are labeled. So the rod is pointing at you and it appears like a single rod, look at the fluctuations of the edge of the membrane and do a Fourier mode analysis and get a fluctuation spectrum which looks like this and get some interfacial tension on it. So, right. So with this, then with this kind of system, you can really do very fun stuff in the sense that so you should think of colloidal membranes as essentially a platform for modern membrane physics. So in cell membranes, it's a heterogeneous system. You have all kinds of lipids and proteins that are floating in the membrane, and then there is this idea that it's not a uniform mixture. You will have phase separation or microphase separation of lipids and proteins within these sheets. But of course, it's a complex system. You can't really figure out why these are phase separating and what stabilizes these clusters and so on. And so here, what we were doing is we were doping short rods in a membrane of long rods. And so these two bright circles are actually clusters of short rods that are embedded in a membrane of long rods. So because of lighting, I think it's harder to see the contrast. And then you can really assemble lots of these clusters, and here it's a dynamic liquid system. So the point of this study was you can have very different kinds of morphologies, you can get remnant phases and so on. So the point of the study was that with this study we could really make measurements on what is the interaction potential between these clusters, why are they forming in the first place, and what is the kinetics of formation and so on and so forth, because everything is stable. So I will not go into the details here. I will now just present to you one more one, which is more directly biophysical in nature. Since this whole series was about physics of living systems and so on, here nothing was really given, but say I took some viruses were not alive, and I self-assembled some structures of them. Right. So this is the work that So this here we were looking at. So now I'm completely switching gears. I'm going to talk to you about freely moving adult cells. And we're going to look at what is called phototaxis, or the cell's name is Femtomonas, and we're looking at collective phototaxis. So this is the work of my graduate student, and we collaborated with colleagues from Prerna Sharma. So here what I'm going to do is I'm going to just introduce the organism and what phototaxis is and what are their collective effects, and then conclude. Okay, so the reason you want to study algae as a particular, normally in conventional biophysics, first thing you will think about is bacteria, right? And because there's a lot of health and disease aspect to it, that you have antimicrobial resistance, you want to understand how bacteria move, how they cause infections. I would call algae as equally important in the following sense that algae have ecological consequences. So when you have marine pollution, you get these blooms of algae, which then further hamper other organisms. So you want to understand how algae come together, and algae are also used in industrial processes to produce drugs and products that are useful for us. So the particular algae that I'm interested in is called Prerna Sharma. It's a model organism. It's easy to grow. Large amount of nutrients are available in the repository. This cell is held on a glass pipette, and it this, right? And we sometimes call this organism as a simple harmonic oscillator of glycolysis. It's literally an oscillator, and we map every problem to oscillator. So here is my version of it. So its beat frequency is 50 Hz, and it swims at 100 microns per second. So it's actually a very big cell. It's about 10 microns. So typically, all these swimming microorganisms, they would swim 10 body lengths per second. So bacteria, since bacteria is about 2 microns in length, it will swim at 30 microns per second. But this fellow being very big, very powerful, it will run 100 microns per second. So you need high-speed cameras to look at them. Because the Tesla is beating at 60 Hz, that means you need to record the motion of this object at least 500 frames per second. And then you need cameras which have very wide field of view so that you can record their trajectories as long as possible. This microorganism, algae in general, they do what is called phototaxis. So if you turn on a blue light, they will run towards that. And there are other examples in nature where you will find this form of directed motion. So it's basically a stimulus response behavior. Stimulus is the light, and then the cell is responding to that light. You have chemotaxis for bacteria or for sperms. Where bacteria will do chemotaxis because it always wants to reach favorable nutrient conditions, and sperm is doing chemotaxis to reach the egg. And so essentially, taxis is a strategy used by microorganisms to reach favorable conditions for life, right? And it's essentially a response to gradients of stimulus. And traditionally, this has been studied by biologists in a very detailed manner, and but in the context of single cell. So if you ask any biologist how does chemical detect light, they will tell you in great detail what are the proteins that detect light, and then what are the downstream signaling pathways that will reorient the cell towards light, and so on. And I think that information is really valuable. But then one needs to take one more step, because in nature there is never a single cell. You always have a collection of cells. And when you have a collection of cells, they in principle can be moved, and we wanted to capture all of it. So as this is mainly what we are really, so that's what I mean by collective phototaxis, meaning I know how one cell will run towards light. Does anything change if I have many cells together? And this too has been studied by biologists in this context of chemical signaling, like quorum sensing, etc. When you say that bacteria are releasing some chemicals and informing other bacteria of the similarities and stuff. So, but I was more interested in the physical aspects of it, and I'll show you how. So, here is a movie so this is a movie where you zoomed out, and each black dot is essentially a single cell. And as I mentioned to you, these cells are light responsive. So you image them with infrared light so that they don't get stimulated by your imaging light itself. Right, red or infrared. And take this movie. So by pitch dark, what I mean is there is absence of stimulus. And you see that all these dots are moving in random directions. When you turn on the light, you see a completely different behavior. You see it as if there is a flow, but actually they are just detecting light and running towards it. If you look at this movie carefully, you see that some of them are running in the wrong direction. And these, you could just ignore this, right? You could pass off and say, oh, something is wrong with these cells, and it's nature, and let's not worry about it. But then we were worried about it in the sense that the fraction of cells that run in the wrong direction depends on the cell concentration itself. So if you have very dense suspension, then you don't have that many AY cells. And if you have very dense, I mean, if your cell having like this, then you have sizes of fraction running in the wrong direction. So clearly, it's not that some cells are something is messed up with them. So now so before I show you the data and I show you the mechanism of it, I need to talk to you about this idea of how does the cell take life. Because ultimately that is what becomes important in this phenomenon. So what this cell has is it has two flagella and it has a single eye spot. So eye spot is a primitive version of eye. So remember, this organism has no neuron, no brain, nothing. But it can detect light and it can move, right? So what is happening is that it has these photoresponsive things that are sitting in the eye spot, and then this cell is doing something interesting. So it has these two degrees of freedom. One is that if you just look at a cell and you find these flagella, I showed you this beautiful movie where the two flagella was synchronously moving. But that synchronous behavior is short-lived. Quite often it will have a phase slip. It will lose that synchronous. And when it loses a synchronous, so if it is perfectly synchronous, it will keep moving in a straight line. The moment you have a phase slip or a loss of synchronous, you will turn. Because it's like, imagine one flagella is beating much more than the other, and it's turn. And the cell will spin about its body axis. So that is the second degree of freedom, that it will rotate about the body axis at some frequency which is called omega. So a combination of this will give you a helical trajectory. So the way what this cell is doing is, if it wants to scan its environment, it has a single eye, so that it has to spin its body in order to scan the 3D environment. Now, you can visualize this rotation with a very unique kind of microscopy. So far I have been talking to you about bright field microscopy. You just shine light and see all the pulses microscopy where things will emit light and infrared and so on. So this is what is called polarization microscopy, where you have polarized light and in which birefringent sample in cross-polarized. So you will get some signal if your system is right. I mean, if the system is uniformly isotropic, then cross-polarized will give you dark, which is most of it. But then this bright spot is actually the eye spot, because the eye spots have this pneumatically aligned fibers of So now I'm imaging the cell in cross-polarizers and I will play the movie. So you can see how the spot goes around. So you can see the cell body location and measure it in this way. So this illustrates how the cell is scanning its environment. The way it reorients itself is the following: that it will, so the two flagella have these names, cis and the trans. The one that's close to eye spot is called cis, and away from eye spot is called trans. And as soon as the light is detected, the cis is suppressed and the trans is enhanced, so that the cell turns, and then it alternates depending on where the eye spot is. And so in that way, it continues to turn towards light. So this mechanism was, figured out by After and Narish in a very, unique way, right? Because in 1995, we did not have high-speed cameras. They were doing stroboscopic imaging. They did not even have video cameras. So they took pictures and pasted it on the walls of their laboratory to figure out the differences in the beating of the flagella. So they held the cell on a pipette and shine light, and then they were imaging the flagellar motion through stroboscopic imaging, and then pasting all these pictures and figuring out the flagellar waveform. Now, modern in the right direction, and the density, the cell number density. So these plots were already there. Our contribution was in trying to figure out that why you have this kind of dependence. So, right. So what I will, so basically what we were doing is, so we knew photocatalysis is concentration dependent, the origin is unknown, the kinetics was unknown, and present-day measurement techniques are now far more advanced. So we did a very simple experiment where we have a 10x objective, we have a known channel, this is our field of view, there is laser that's shining here, and we are watching this object. So, I mean, these cells are actually incredibly sensitive, right? Initially, we were just novices fooling around with these cells, and so we take these chambers and just keep it on the, on a table, and you have lights on. They will do phototaxis. In the sense, they will tell you which light is brighter. So, yes. Right. You mean frequency of light? Yeah. So, I mean, all these cells have variable dependent behavior. So, these cells respond to blue light, and they do what is called positive phototaxis. They run also. Then, if you shine green light, they can run away from it. So, it can become negative phototaxis, for example. So, there is variable dependence both in the sign as well as efficiency and so on. But largely, think of them as plants. That's why they are most responsive in UV region. Yeah, and so then, we start. First thing that you need to do if you have a suspension is you characterize it in the dark. So, cells are moving randomly. What I'm plotting here is mean squared angular displacement versus time. So, we are familiar with mean squared displacement, which is the translational degree of freedom. Now, because these cells are anisotropic and have directionality, you should think of the orientation as well. And so, this is a diffusion in the orientational variable. And the rotational diffusion coefficient is given by the slope, and in the range of the concentrations we were, this diffusion coefficient was constant. So we did not have a concentration dependence for this rotational diffusion coefficient. Now comes the data. So now we turn on the right, and this is the movie that I showed you earlier. Now what you can do is, so this is our data, right? So we write a lot of image processing programs to track each of these plots and build a time series of the position of each of these cells. And then you can plot these trajectories. So these trajectories are colored arbitrarily. This blue arrow is the direction of the light. So in the movie, light is here, in the plot, light is here. And what you can see is that a large number of them are running towards light, but then there is a whole lot of other trajectories that are going in random directions. So all of these trajectories were like, set to 0, 0. So this is the starting point of each trajectory, and you can see that they are going in one direction, all possible directions. So you can essentially say, okay, let me find out fractional trajectories that are within some 5 degree or 10 degree of the right direction, and plot that as a function of highly invincible cell. So the stronger the signal, the higher the efficiency. That's what you would expect of a sensor, right? You can plot the probability density as a function of angle. And so black is pitch dark in the stimulus, then blue is weak stimulus, red, green is stronger, and green is the highest stimulus. And you can see that the probability density very nicely increases. So now let's look at the data, which is as a function of concentration. So we hold the light intensity constant and change the cell concentration. And you can see that in dilute conditions, it is actually it's pretty good. Then at some intermediate concentration, there is a lot of confusion, and then at higher concentrations, once again you have recovery towards the high alignment. And so here are the probability density curves that are offsetted for parity, and you can see what I was describing saying in words that the peak height goes down and then goes back up. And you can plot what we call as phototactic efficiency, which is fraction of cells that are running in a small angle of the y direction. And what you can see is that at low light intensities you have this re-entrant feature that the efficiency goes down, then goes back up, and then this effect becomes bigger at higher intensity. So later on, so this data initially was at 4 different concentrations, or actually 10 different concentrations with 2 high intensities, and later on we did it for many different intensities and set concentrations and had a similar behavior. So I mean these data sets are, when you are working with living systems, that can be a real challenge, right? So we have like and plotting data pulled from like at least tens of trials and millions of trajectories, right? And so that's the only way you can kind of get some understanding. So look how big the error bars are. And normally in a physics experiment you will be like, you look at very small error bars. Sometimes your error bars are very simple size. And so when you have these large error bars, your kind of heart sinks, okay, this is a really noisy system and you really do a good job of getting data. So how do you model? Yes. Yeah, so I will come to that. That is the full, that is my story, that what is causing this decay. So, how do you model something like this? You say that cell is like an active grounding particle, and your live field is like a curve that aligns the cell along the right direction, and you can write down very simple equations of motion for the position and orientation of the cell. So the position, so this is the fact that the cell is running at some constant speed, and this is the thermal noise, and the orientation is undergoing this, it's in presence of this external potential due to the light, and it's angle dependent, and you can write it in this way. Gamma is the strength of the torque, and then you have the rotational noise. So you can then, when you have a collection of such cells, you will write down a Popli line kind of expression and get a expression for angular probability density, what we were plotting, and this is what the analytical solution is. So in this case, it's actually easy to get the analytical solution. So this is your standard differential, right? dr as per psi, and then you have another term that is because of this external potential, and that has this derivative dependence on it. So, so we can fit. So this is basically looks like a Gaussian function. So we can fit our experimental data with this Gaussian function, and the width of this function is the ratio of the strength of the torque, the rotation and efficient rotation. So now it gives us a handle in the sense that you can fit data and get some estimates for the torque, and that torque has the same dependence as the efficiency. So I haven't really, in these equations, I haven't really included any collective effects or interactions. This is completely being modeled. But it's a good exercise in the sense at least you have a sense of what would these terms would look like in completely non-interacting setting. So now comes this part, that why are they, why do they have this kind of re-entrant, or why does the efficiency improve with increasing concentration? So as I mentioned that initially we thought maybe it's like photon sensing. When I have lots of neighbors, they're excreting some chemical and guiding others towards that. But we didn't really have good evidence for it. And then since the photon sensing idea fell apart, then we started to look at physical mechanisms. And one of the ways the physical mechanism becomes important is that the average speed of cells comes down when you increase the cell concentration. Basically, you have a very crowded environment, so you will move slower. So here is the mean speed of cells as a function of cell concentration, and then there is some decline in concentration, and we are plotting data above this critical concentration. So by critical concentration, what I mean is this concentration. So I am not really exploring this crowding effect, what they say, think about it. So how does slowing down help? Why would a decrease in speed improve phototaxis efficiency? And it goes back to how the cell detects light. So remember I mentioned that the cell is always rotating about its body axis. Now the rotation frequency around the body axis is coupled to the linear speed. And we can measure the rotational frequency also as a function of concentration, and that also had a similar impact, right? So now what is happening is that when I have more neighbors, so I'm in a dense suspension, I am turning slowly, and if I turn slowly, then I'm going to integrate my stimulus much better because I'm facing the light longer, and therefore I have a better shot at making a decision where the light is and how to turn. And in this way, the efficiency improves. So it basically enables the cell to collect more photons per unit time and detect the light direction more accurately. So if this hypothesis is true, then what I should do is I should, instead of changing the speed by changing cell concentration, I should hold the cell concentration constant and increase the viscosity of the media, and that will also slow down cells. So here I've added some polymer, and I can reduce the speed by increasing the concentration. So now what you do is you plot the phototaxis efficiency against mean speed. So you stop worrying about whether I have increased the cell concentration or I have increased the viscosity of the media, and you find there is a very nice correlation. So the green points are the points that we took by changing the cell concentration, and magenta points are the points that we took by putting cell concentration constant and increasing the viscosity of the media. And so then this is like, I'm very happy that this was a very nice story that tells you that you will do much better in life if you slow down. So with this I will stop and take questions.
In that region, because you do see a negative correlation and then you see a positive correlation.
So I mean, so yeah, that's why we were doing this experiment in a really long that and very far away from the light. So cells are themselves very sensitive. When you do it in a long chamber, you can hold cell concentration more or less constant in your image. Otherwise, yes, I mean, if you were doing experiment here, we are in deep trouble.
If I understand your story right, then when you do whatever is necessary to decrease the efficiency of phototaxis, it should affect all the cells in more or less the same way. But
yes.
You are finding that some cells seem to be more susceptible than others. So there's something else going on at the level of difference between cells. So what I would have expected from your story is that each cell would lose its way more often and then find its way. Rather than your trajectory seems to show that some cells are totally lost and some are just really straight.
See, I mean, what I'm trying to say is that it's not that the cells that are lost, they're lost because there's something wrong with them. Basically, your stimulus is very low, and you have a detector which has some efficiency of detecting that stimulus, then a fraction of those will always make a mistake, right? And now what I'm saying
So
is,
there's some difference in threshold.
yeah, there could be slight differences in this threshold of detecting. And then what I'm saying is you can rescue this behavior by slowing them down, and then they will all be fine. So my story is that there's nothing genetically different about them. I mean, okay, there may be, how should I say, there is something different about them, but it's not in the way that the cell is, its DNA is a food for them. Yes.
Why we see this random behavior in the mid concentration only? We have not seen this in very low concentration or in very high concentration, but in the mid concentration we have seen this random motion that they are going away at these things. Why is that?
Right. So then again goes back to so this block, right? So so you can say that the efficiency, essentially phototaxis efficiency, becomes quite low as the speed increases. So at this intermediate concentration, the speed is pretty high. That's where you are sitting here, and your rotational frequencies, you're not able to integrate the signal fast enough. So the moment you start integrating your signals longer, then you make less mistakes. So there is basically, okay, the way to also look at it is, there is a critical concentration at which this phenomenon starts to kick in, right? That notice the speed is kind of flattening out. So at lower and lower concentration, that's the speed that you have if you had isolated cells. Then there's a critical concentration above which things start to have this density dependence. So there's a density dependent motion that in dilute to semi-dilute conditions, your average speed of cells stays constant or independent of cell concentration. Then above a critical concentration, the speed starts to dramatically go down. Yes.
When you were like deciding, when you were giving the light as a torque, right? Like you would expect a torque to act on each thing the same way. Expect each
Right.
cell to be rotated the same way,
Right,
but like that's not the behavior you observe. So then,
Right.
how do you model that as a?
So as I said, that model was completely, it did not encode any connective effect. What you would have to say is that the torque itself will become number density dependent. So torque will become cell concentration dependent, and then you need to have a model for it, right? Does it depend linearly on concentration or quadratically and so on and so forth? And that's something that we couldn't really come up with, like a model that would motivate that makes sense. So we were trying to do these kind of measurements that we said, okay, they're not just scalar coupling, you can have hydrodynamic coupling, because after all they are swimming in a fluid. Let's put tracers, let's measure flows, and then say whether flow field of one cell impacts other and so on. But we didn't make much progress with it, because those models can be even more harder to hook up and validate with data. So that's what it is. Okay? Yes.
Isn't it light spot through evolution has made the modern eye?
What do you mean by that? Like,
This simple light spot
Yes.
which you showed in
Yes.
environment.
yes.
So by evolution is that resulted in the modern eye that we see.
Yes. In the sense, that's the most primitive eye you could think of. In the sense that these proteins were detecting light and causing downstream changes in the cell body. In this case, right? What are our bodies doing? We have some proteins that detect light and then send electrical impulses to the brain, right? So then it's a similar idea, but of course executed in a very different set of proteins and machines.
But it results with evolution, same thing.
I don't know why everything is evolution in the sense we have evolved from there. I will not say that this is a good model system for mammalian animals. We are all mammals, right? And this is not the model system for mammalian animals.
I think another way of putting the question is there could have been many ways in which our modern eye could have begun in the ancient
Yes.
past. Is this the beginning that led to this eye, or is this Does this go somewhere else?
I don't have a knowledgeable answer. I will refrain from yes.
How do the flagella work?
How do the flagella work? That's a very good question. So flagella is again made up of microtubules and motors. Okay. So I actually tried to explain that yesterday. I'll remind you. Or maybe now I will make use of the presentation and just show you the schematic and maybe then it will make more sense. So unfortunately I don't have the talk opened up, so I don't have so here is let me try to explain. Okay, so this is the flagella and this is the schematic. So you have a plasma membrane or cell membrane, in which you have these 9 microtubule doublets. By doublets, what I mean is 2 microtubules kind of fused together, and then between the 2 doublets you have the molecular motors. And what these motors are doing is, the job of motors is to actually slide the film. But now, all the whole assembly is cross-linked. So there are lots of cross-linking proteins between each of these doublets. So since the motors cannot really slide them, they will apply local forces. And so the tangential force, because it's cross-linked, this tangential force will give you a normal component. And then that normal component will bend the film. And there is it's a big question in biology as to how the cooperative action of motors bring this beautiful waveform. In the sense that if you want to, so this is like a cylindrical arrangement of motors. So motors on one side are activated and they will bend the filament this way. But once it has reached its extreme, then this side of motors have to be given and this side of motors have to let go. And so that the filament now will bend the reverse way. So there is this intricate cooperative switching mechanism that happens autonomously in this system, and you get this. Since you brought me to this slide, I will just show one more slide that I think was one of the most beautiful experiments we had done, which was this. So what we did was we took these cells and we cut the flagella out. And the reason we wanted to do that is because the flagella is being on a very big cell body. So as a boundary condition, it's very difficult to handle when you are trying to measure fluid friction and things like that. So we cut it, and then because these are motors and they consume ATP and do work, so when you add ATP, these filaments will actually show. So it's like cutting someone's arm and still having it to work, right? Because you have a motor microtubular assembly, here it's freely swimming. Here I have clamped the filament, and look how it's beating. It's beating just the way it was beating on the cell. So what this tells you is that just the mechanochemistry of motors alone is enough to give you a spontaneously moving oscillator. You don't need the whole cell machinery to do this. So, and then we put tracer particles, special flow fields, and lots of things to say interesting things about it. That's really good. So,
The cell can just supply differential ATP and then get the phase difference.
yeah, cell is just supply data. That's it.
Experimenting how challenging is to perform all this. What kind of equipment are needed? Like for clamping
Actually, clamping is easy. It's just you modulate, the surface conditions of the cover slip when you do it. This actually, all that we needed was a high-speed camera and a microscope and light source and optical cable because we want to isolate it from the flow vibrations inside. So as an instrument, instrument point of view, I will just make this comment that this kind of science is really tabletop. You can just go into a room, you have a table, you have a microscope, then that's it.
expensive table and a very
Yes,
expensive
all I can tell you, absolutely, the whole setup will cost 1 crore. Okay, 1 crore is in my house. But I would just like to remind you that if you go to a low temperature lab, you will have a dilution fridge hanging from the roof or going into the water from this earth, and that itself will cost 4 crores, and then there is electronics, etc. The whole thing is just
comparatively cheap.
is comparative.
Anything progress is expensive.
Now, how long this survives without the cell phone?
And sorry,
How long this plasma survive without the cell phone?
it will go as long as ATP is there. So we have these ATP regeneration systems because it's the fuming ATP, right? So you need a cyclic reaction to maintain ATP constant. So with that feedback loop, we can see up to 40 minutes or so. But if you come and you add more ATP, it will go on. I think it may last. Okay, I should be honest a bit here. What happens is there is a chlorine. So your buffers will, no matter how much you autoclave or standardize, etc., they will get some enzymes which will eat these proteins. So after 24 hours, it will not. Because the machine will get damaged due to these incubations. But it's a beautiful molecular machine. And this is the beating that our cells do. I mean, so this filament, actually a large number of these filaments line our lungs and push the mucus out. So literally we breathe because they beat. They stop beating here. Okay, I'll stop here.
This is really today what I have observed is, not only we have like classmates today, two professors basically, they are both PhDs, and they are basically, shouting, arguing, and so much, sort of discussing this, harshly in the corridors of the schools. And one was basically a biologist, and the other one is a physicist. And they both basically have their completely independent thought that one is showing this simplicity that we are the only one who can describe the other one, we are the one actually who understand this life more than this people. This is actually happening at the time. Obviously, this kind of work which we have started much earlier, but because of the information is not available as it is today, maybe they are not aware of that. That one day they might not have thought that one day basically a physicist is trying to basically explain certain part of the biology. Obviously, there are still actually far away at one point actually they can come together and explain how things work, but still basically we are actually moving ahead. And today actually I'm updating the list of my speakers basically. I'm like basically adding basically or Prerna name basically to the list. And I actually just go through this and see that in the past 10-12 years, we had actually 4 or 5 basically series on basically physicists actually going into the biology explanation. First by the at that time I don't remember this part. Then actually Sanjay Jain, Shwan Chakravarti, last year we have Gautam, and today we have this. So I think we also basically together, it is not intentional, let me tell you, but to some region basically we are also moving, to understand life and applying physics to that. So thank you again, Prerna. Thank you to my colleagues for coming here, actually doing it, plus basically the idea to choose the right person basically for this talk. Thank you to all of you. Without you people actually this series basically, because it is only for you people basically we are actually arranged. Thank you all. And in the last, I hope that we should basically get to know the people actually who's working for this event basically finally begin and come to the campus and the Physics Society member. Please come quickly and come to the these are the people actually who are working actually day and night for this event. I think you should have
अरे सामने आ रहे हैं